A few hours from now, your body will be paralyzed. Not sort of. Completely. The muscles that move your arms, your legs, your jaw, all of them will disconnect from your brain. A small cluster of neurons in your brainstem will send a signal down your spinal cord that shuts off every motor nerve below your neck. It will happen 4 to 6 times tonight. It has happened every night of your life. It is not a malfunction. It is what keeps you from physically running into your bedroom wall every time you dream. The phenomenon has a name and a moment of discovery. In 1962 at the University of Lyon, a neurophysiologist named Michel Jouvet, pronounced Jouvet, was trying to answer a simple question. During a certain stage of sleep, he could see on the EEG that a cat's brain was firing like it was awake, and yet the cat was not moving. Something was holding the body still. Jouvet lesioned a small region deep in the cat's brainstem, the pons, roughly the size of a fingertip. Then he ran the animal through another sleep cycle. The EEG still went into the active state. The eyes still moved. But now, inside the lab, the cat stood up on all fours. They hissed. They stalked invisible prey. They groomed cats that were not there. He called the stage rapid eye movement sleep, REM. And he had just proved, in the simplest possible way, that the dream is something separate from the body, held apart by a small piece of brainstem. Here's how the same system works in you. Step 1. As you enter REM sleep, a small cluster of neurons in your pons, in a region called the sublaterodorsal nucleus, often shortened to SLD, starts firing. The SLD fires only during REM. At every other moment of your life, it is quiet. Step 2. Its axons travel a few centimeters downward into the medulla. There, they hand off the signal to a population of inhibitory interneurons. Step 3. Those medullary interneurons send their axons the rest of the way down into every level of your spinal cord. They release 2 neurotransmitters onto every alpha motoneuron in the ventral horn, glycine for fast inhibition, and GABA, pronounced GABA, for a slower tonic background. Step 4. The motoneurons hyperpolarize, motor commands arriving from your cortex to swing a fist, to kick, to speak, still reach them, but the motoneurons no longer fire. The muscle receives nothing. Your brain is awake, your body is offline, and the biology built a redundant brake because one transmitter alone is not enough. You probably think of paralysis as something that goes wrong. A stroke, a nerve injury, a disease. The idea that your brain actively paralyzes your entire body on purpose every night sounds like a category error. Think about what happens without it. During REM, your motor cortex fires almost as strongly as when you are awake. The dream is not a gentle slideshow. It activates vision, emotion, and motor planning in a sequence your brain treats as real action. Without a safety system, those motor commands would reach your muscles unchanged. You would stand up and try to run. So the biology built not one safeguard, but 2. Glycine flicks each motoneuron off, one by one, moment to moment. GABA sits underneath as a slower, continuous background. In mice, if you block one of them, the animal shows partial dream enactment, a leg kick, a jaw clench. If you block both simultaneously, the atonia fails completely, and the animal acts out the full dream. 3 things are allowed to keep moving. Your diaphragm, which runs on a separate respiratory circuit, keeps breathing. Your extraocular muscles, wired differently, produce the rapid eye movements the stage is named for, and 2 tiny stabilizers of the middle ear. Everything else for the next 10 to 20 minutes at a time is clamped by a brainstem you will never consciously feel. The mechanism was mapped first in cats, The break was documented first in humans in 1986. At the Minnesota Regional Sleep Disorder Center, a neurologist named Carlos Schenck, pronounced Schenck, began seeing a series of elderly men referred for violent behavior during sleep. One had repeatedly punched his wife while dreaming of intruders. One had dived from the bed and broken his nose on a nightstand. One had leapt from a second-floor window, convinced he was being pursued. Schenck brought them into the lab and ran overnight polysomnography. The EEG showed unambiguous REM sleep. The EOG showed the rapid eye movements, but the EMG, the signature of muscle activity, was not flat. It was firing in bursts. The motor commands of their dreams were reaching the muscles. Schenck named it REM Sleep Behavior Disorder, RBD. The defining feature on the recording is called REM sleep without atonia. It is Jouvet's cat experiment playing out in a human bedroom. For 30 years, RBD was treated as a parasomnia, strange, disruptive, a problem for the patient and the spouse, but not much more. Then the follow-up studies came in. Schenck went back to his own original cohort 10 years later, in 1996, and counted. Of his 29 men, 11 had developed Parkinson's disease. Then Alex Iranzo, pronounced Iranzo, at Barcelona extended the follow-up beyond a decade, and the conversion rate climbed above 80%. Then, in 2019, Ron Postuma, pronounced Postuma, at McGill pooled 1,280 patients across 24 centers in 12 countries, the largest RBD cohort ever assembled. The number settled around 73% neurodegenerative conversion within 12 years of diagnosis. Of those converters, about 43% developed Parkinson's disease specifically. The rest developed dementia with Lewy bodies and pronounced Lewy, or multiple system atrophy. All three diseases share the same underlying pathology, aggregated alpha-synuclein, pronounced synuclein. The median lead time from the first dream enactment symptom to clinical Parkinson's is 10 to 15 years. The neuropathologist Heiko Brock, pronounced Brock, had proposed in 2003 that Parkinson's pathology begins in the lower brainstem and ascends. The dopamine cells of the substantia nigra, the ones responsible for the motor symptoms that define the clinical diagnosis, are hit relatively late. The SLD region sits directly in the earlier stages of Brock's map. When atonia starts to fail, it is telling you where the pathology has reached. None of this explains why alpha-synuclein chooses the brainstem first, why the SLD region is hit before the substantia nigra years before, why the disorder strikes men 4 or 5 times more often than women, why roughly a quarter of patients with idiopathic RBD, followed for a decade and more, still do not convert. There's also the question of what to do. Clonazepam and melatonin at night can reduce the behaviors, keeping a partner safer and a patient in bed. Neither changes the underlying trajectory. Several trials are now testing whether alpha-synuclein targeting drugs, ambroxol, nilotinib, monoclonal antibodies, can slow the conversion in people diagnosed with idiopathic RBD before any motor symptoms appear. If any of those trials succeed, they will succeed first in these patients because they are the earliest identifiable. A window of 10 years is not a long time, but it is the longest window we have. You are paralyzed every night, and it is saving your life. A small circuit in your brainstem holds down a body that would otherwise be acting out every chase, every fall, every fight your mind dreams up. You cannot train the system. You cannot feel whether it is working. But if a partner tells you that you kick or punch or shout during sleep, that is not a quirk. That is a neurology-grade observation about a brainstem structure, and it deserves a neurologist, not a shrug. Every week, another piece of the machinery that runs your night. Subscribe.